Let's go ahead and get started. Uh, does that work? Yeah, I think that works. Okay. Okay. Neurophysiology, Chapter 3. Electrical signals are the vocabulary of the nervous system, not chemicals. It's an electrical it's an electrical signal. Molecules that maintain an electrical charge are called ions. And let's take a quick look at this. Maybe. There we go. This is out of the University of Utah.
Signals from our environment are most often first detected by the brain. When someone is frightened, a stress response is initiated by their reaction to an environmental signal. But many signals don't stop here. A signal received by the brain can be passed on to many types of cells throughout the body and have widespread effects. Signals get sent around the body by traveling through the nervous system or bloodstream, going from cell to cell. You could think of the cell's sole purpose in life as keeping signals going or initiating new ones in response to the environment. Once a signaling molecule from the bloodstream reaches a cell and docks, the signal is passed into the cell. Like a miniature mousetrap game, the cell is filled with complex contraptions called proteins that bump and nudge each other to keep the signal going until it reaches its target. In this case, it's the production of yet another signaling molecule that will get released into the bloodstream, dock on other cell types, and cause them to react in a variety of ways. The signal-passing machinery inside the cell can look and work very differently, depending on the intended effect. Here we see a signaling cascade that results in the release of energy molecules. Once liberated, they are sent on their way via the bloodstream to power cells in another area of the body. When this tiny domino effect is triggered inside cells, some amazing things can happen. Other signaling cascades triggered during the stress response result in a variety of physical reactions. This signaling molecule can relax or contract different types of muscle cells. When it reaches the skin, the signal docks on muscle cells attached to a body hair. The tiny muscle is stimulated to contract, raising the hair. The signal also docks on cells wrapped around a sweat gland, which contract too, squeezing sweat onto the skin's surface. In the lungs, the signaling molecule docks on muscle cells wrapped around bronchioles. But these cells relax instead of contract to allow more oxygen into the blood. Because the protein machinery inside each kind of muscle cell is different, this single type of signaling molecule can cause both relaxation and contraction. Many signaling molecules work this way. When this signal reaches the heart, it docks on beating heart cells. Their response is to beat faster. Blood pressure goes up, and the signaling molecule is rushed on to still more cell types around the body. Constantly and in concert, Cells must send and receive a myriad of signals. Cell communication is vital to keeping the body alive.
Ta-da! Okay. There we go. Okay. Neurons contain large complex proteins that maintain a negative charge, but because the channels are closed to the larger positively charged sodium molecules, only the smaller positively charged potassium ions can pass through the small channels. Because of the large negatively charged proteins, a neuron at rest maintains a negative resting potential of from negative 50 millivolts to negative 80 millivolts with an average of negative 70 millivolts. Negative 70 millivolts is the potassium equilibrium potential, as equated by the Nernst equation. There are many more potassium cations inside the cell than outside, while there are more sodium cations outside than inside. When the neuron is stimulated by a neurotransmitter detected by a dendrite, the chemical neurotransmitter causes localized depolarization. In other words, the neurotransmitter causes the sodium channels to open in the immediate area. For a neuron to react, it must receive an adequate amount of neurotransmitter to stimulate enough sodium channels to open up. This is referred to as the neuron's threshold, usually a depolarization of negative 50 millivolts. If the threshold is reached, then enough sodium channels will open to trigger the depolarization. A neuron cannot partially fire, it either fires or it doesn't. This is known as the all or nothing property. When the threshold is met, sodium ions flood into that portion of the cell through the rapidly opening sodium channels. This triggers the sodium channels next to the stimulated area to depolarize as well, causing a chain reaction along the neuron until it reaches the neuron's own synaptic cleft. And another video from YouTube. Yay! Sounds like crickets. Good.
Unstimulated, neurons maintain a constant electrical difference or potential across their cell membranes. This potential, called resting potential, is always negative inside the cell and ranges from negative 40 to negative 90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative depending on the stimulus. If potential is made sufficiently less negative, it reaches a level called threshold, and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell restores its negative resting potential. The cell membrane of a neuron encloses cytoplasm with various ions dissolved in it. The neuron itself is immersed in a salt solution, the extracellular fluid. The ions of the cytoplasm consist mainly of positively charged potassium ions and large negatively charged organic molecules, such as proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up cell membranes, they must travel through channel proteins that extend through the membrane. In an unstimulated neuron, only potassium ions can cross the membrane, traveling through specific proteins called potassium channels. Although sodium channels are also present, in unstimulated neurons, they remain closed. Since only potassium ions can cross the membrane, and potassium ions are more concentrated inside the cell, they diffuse out of the cell, leaving the large negatively charged organic ions behind. As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside. At some point, the diffusion of potassium ions out of the neuron due to concentration differences is balanced by the electrical attraction tending to pull them back inside. This is the point at which neurons reach resting potential. Reaching resting potential in this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of the potassium ions initially inside a neuron must leave to create a resting potential of negative 60 millivolts. But it is action potentials, not resting potentials, that carry information through a nervous system.
All right. Action potential refers to the amount of positive ions that are allowed to flood into the stimulated area of the cell through the sodium channels before the sodium channels close. The action potential for most neurons is around plus 40 millivolts. When enough sodium ions flood into to reach the positive 40 millivolts, the sodium channels will close. Going beyond equilibrium with the outside environment at 0 millivolts is called overshoot phase. While the neuron is recovering from stimulation, it cannot be stimulated. This is known as the refractory phase. During the refractory phase, the sodium channels have closed, shutting off the supply of sodium ions flooding into the neuron, and potassium channels will open. Because the electrical charge outside the cell is more negative than inside the cell, potassium ions are drawn out of the cell. With the positive potassium ions now flooding out of the cell quickly loses its positive charge, and it not only reaches its resting phase potential, but it goes beyond it in a phase known as the undershoot phase. The potassium leak channel now allows enough potassium ions to re-enter the cell to return to the area to its resting potential. Neuron dendrites and somas usually have a different type of membrane than axons. Dendrites and somas usually have sodium channels that are stimulated by chemicals and not by voltage. In other words, they're only activated when the chemical neurotransmitter is present. The axon, on the other hand, does have voltage-gated sodium channels, so the axon responds to stimulation from the dendrite and the soma. Neurons in the central nervous system are myelinated by oligodendrocytes, and neurons in the peripheral nervous system are myelinated by the Schwann cells. These are the glial cells that we talked about before. The myelin covering of the neuron does not allow potassium leakage from the area covered by the myelin. The depolarization voltage is channeled down the axon from one node of Ranvier to the next, increasing the response by as much as 15 times. This form of conduction is known as saltatory conduction. And you can see the nodes of Ranvier here. These are the nodes of Ranvier that they were talking about. Multiple sclerosis is an autoimmune disease where, due to a genetic flaw or environmental trigger, an individual's immune system destroys the myelin on select neurons. This causes the slowing of motor movements and the loss of sensory abilities due to the slowing of neuronal response. Most synapses are triggered by a chemical neurotransmitter in the synaptic cleft. But researchers have discovered that some synapses in the brain are stimulated by electrical responses from another neuron. This is able to occur because the synaptic cleft of these neurons is 1/10 the distance of a chemical synapse. Not all neurons cause their connecting neuron to fire. Some neurons are inhibitory neurons, and instead of raising the excitatory potential, depolarizing the neuron, it lowers the excitatory potential, hyperpolarizes the neuron, making it more difficult for the neuron to fire. Inhibition is done by inducing channels in the receiving neuron to open, allowing more negative chloride ions into the neuron. Since the chloride ions have a negative charge, they lower the neuron's potential to fire. An inhibitory or excitatory neuron is determined by the neurotransmitter they produce. Some neurons have hundreds of other neurons feeding into them. With these neurons, they may require a number of neuronal stimulations before their threshold is reached. This accumulation effect that is required for stimulation is known as spatial summation. Spatial in this case meaning the amount of space that they're taking up that require the stimulation in order to fire. Some neurons that require an increased threshold will not react from a single neuron stimulation. However, if that single neuron fires in rapid succession, the accumulative effect of the neuronal stimulation will stimulate the neuron to fire. Because it is a matter of adequate stimulation in a select time period, this type of stimulation is called temporal or time stimulation. Since a threshold has to be achieved before a neuron will be stimulated, there are various structural factors that can increase the likelihood that a neuron will be stimulated. The number of dendrites receiving stimulation increases the probability that the threshold will be reached. If a glial cell overlaps a synaptic terminal, it may increase the amount of neurotransmitter in the cleft by not allowing leakage. A chemical synapse works in the following manner. The electrical charge travels down the axon to the axon terminal. The charge changes the permeability of calcium channels, allowing calcium to enter the synaptic terminal. The calcium causes the synaptic vesicles to fuse with the terminal membrane, releasing the chemical neurotransmitter contained in the vesicles in the synaptic cleft. Now we've talked about calcium, we've talked about sodium, we've talked about potassium, we've talked about chloride. These are all chemicals that are very important to the functioning of the human body. And this is one of the reasons why if you tasted blood, it tastes like it's salty. If you taste any fluid coming out of the human body, it tastes salty. The reason it tastes salty is because sodium chloride is very important to the functioning of a properly functioning human body. So it has to have sodium chloride. Also has to have potassium chloride. But we don't have to take in potassium chloride because it's the sodium that's more important. It's the sodium that makes things fire. We also need calcium. If we don't have calcium in our systems, then our neurons will not function properly. And that's one of the reasons why we take calcium into our systems in order for our bodies to function properly. So calcium and sodium chloride and potassium, potentially potassium. Some people don't get enough of these chemicals. They don't get enough salt. And they have a fairly serious problem. We get a lot of calcium in most of our foods. We get salt in most of our foods. But if we had a sodium poor diet, sodium, especially a poor salt diet, then potentially we might have problems in the future. We usually get enough potassium in our food in order to maintain ourselves. But some individuals have an imbalance in their bodies otherwise, so they have to take in extra potassium, excuse me. Okay, so all of these chemicals, sodium chloride, potassium, and calcium are very important. The other chemical that is very important, the other metal that's very important, chloride's not a metal, but the other chemical that's important is magnesium. Okay, so what are we doing? Okay, So if it wasn't for calcium, the neurons wouldn't fire because it's the calcium channels that allow the neurotransmitters to come out of the synaptic terminal. After the release of the neurotransmitter into the synaptic cleft, the chemical migrates to the receiving neuron and attaches to transmitter receptors that either inhibit the potential of the receiving neuron or stimulates a neuron to reach its threshold and fire. Much more neurotransmitter is released in the synaptic cleft than can or will be used. It's an all or nothing situation. It just releases as much as it has, and there's a lot more than it really needs to fire. Getting rid of excess neurotransmitter in the synaptic cleft, since neurotransmitter left in the cleft will cause spasming of the neuron, it must be removed from the cleft. The amount of neurotransmitter is detected by an autoreceptor and triggers a stop to the release of the neurotransmitter. Removing excess neurotransmitter from the synaptic cleft is done in one of two ways. Either an enzyme in the cleft breaks down the excess neurotransmitter, or receptor sites on the transmitting neuron bind with neurotransmitter and reuptake the neurotransmitter. Neurotransmitter that is taken back into the neuron through reuptake is recycled by the neuron. And this is what happens with serotonin. That's one of the reasons why people who are depressed they need extra amounts of serotonin in their synaptic clefts to keep from being depressed. So what we give them is a medication that blocks the reuptake of the serotonin, putting more serotonin into the synaptic cleft. And this chemical that we give them is called a selective serotonin reuptake inhibitor. It inhibits the reuptake of serotonin selectively. SSRIs, and that is the antidepressant that is most frequently used in the United States anyway. That's the one that works that way. Selective serotonin reuptake inhibitor. So there's two ways to get rid of the neurotransmitter. One is for a chemical to enter the synaptic cleft and change the chemical structure so it's no longer a neurotransmitter. And the other way is for it to be taken back by the expelling neuron, and that's the selective serotonin reuptake inhibitor. It inhibits the reuptake. No, okay, well, anyway. This is in German. So that means canal, this means receptor, and that means ligand. The neurotransmitter binds to a receptor molecule on the receiving neuron. Each neurotransmitter or ligand has different receptor molecules that they bind to called a ligand binding site. They are bind to, anyway, that's not proper. I need to change that, don't I? So it attaches to a binding site. This is the binding site here. As you can see, this chemical structure fits into that slot right there. This is a W slot, and it fits into this W slot. Perfect. When an excitatory ligand is bound to a receptor molecule, it opens up a sodium channel in the receptor molecule, allowing sodium to enter the neuron. If enough receptor molecules are stimulated by an excitatory ligand, the neuron's threshold will be reached and the neuron will respond with an electrical charge. If the neurotransmitter is an inhibitory ligand, it will open up a chloride channel in the receptor molecule, and chloride will enter the neuron. So one, it allows the electrical charge to be created because it is an excitatory ligand. The other actually opens up chloride channels and makes it more difficult for the neuron to fire. It puts excess amounts of chloride in the neuron. Neurotransmitters are chemicals. While each receptor molecule binds exclusively to their neurotransmitter, there are similar chemicals that are structurally similar to the neurotransmitter that can bind to the receptor molecule as well. Now, this is really important because this is one of the ways that we can control whether those neurons are going to fire or not. If they're firing too much and they're causing you to have high blood pressure, we can give you a medication that will block that neurotransmitter, and it will not allow the heart to beat as rapidly or for the blood pressure to increase as much. Uh, there you go. That's the way to control things. So we have different chemicals that look like the neurotransmitter that we can give you to control whether your neurons are going to fire or not. Acetylcholine is an excitatory neurotransmitter, but the poisons curare from a South American plant and bungarotoxin from the Bungarus snake of Taiwan bind to the receptor molecule, causing paralysis. Acetylcholine receptor molecules are also the targets in the case of the poison muscarine from the Amanita muscaria mushroom. And this is what the Amanita muscaria mushroom looks like. You potentially have a picture of these guys in your home. I've seen lots of pictures. It is quite an attractive toadstool, I'll have to admit, with all the white and the red and the white polka dots. Muscarine, however, binds to the receptor molecule and mimics acetylcholine, causing the neuron to fire, resulting in spasms, and then you go into seizures, and then you die. This is one of the ways that there are lots of poison mushrooms out there. The Amanita mushroom is one of them, but people have been trying to use toadstools for years as food. And sometimes you get the good ones, but by golly, it only takes one time, and all of a sudden, you've got a problem. Chemicals that mimic neurotransmitters and cause a similar reaction are called agonists. Chemicals that mimic neurotransmitters and block the action of neurotransmitters are called antagonists. And here are a group of nurses telling you exactly that. I hope. There we go. They're still there. That's good.
Our presentation is on agonists, antagonists, and receptors. A receptor is a chemical structure on the outside of a cell where an agonist or antagonist binds in order to send the cell a message. An agonist is a molecule that can activate a receptor to send the cell a message. An agonist is non-competitive, binds irreversibly, or competitive, binds reversibly.
Hi, we're talking about receptors. A receptor is like a keyhole in a door, and an agonist will bind like a key and then go into the door, and you will be able to go inside, like a message.
Antagonist, a molecule that binds to the receptor to block the agonist or antagonist and stop the message from being sent to the cell.
Hi, an antagonist is like the wrong key that will go into the keyhole, but it will not turn. Therefore, the message cannot be sent. I guess we're at the wrong house.
Okay, this information is important for nurses to know because you may be giving drugs that can act as an agonist, which will promote a physiological response to continue, or you might be giving an antagonist, which will make a physiological response stop. An example is when a person has an allergic reaction, they're producing histamine, which is the agonist, and you want to give them an antihistamine, which is the antagonist and stops the allergic reaction.
Achoo!
Here's some Claritin, which will stop that allergic reaction from happening.
Thank you, nurse.
You're welcome.
Oh, University of Texas at Ashland. Okay, way to go. Thank you, nurse. Anyway, antagonists and agonists. I hope you understand it now. An agonist actually makes something happen, and an antagonist stops something from happening. So an antagonist would fill the receptor site, but it would block the neurotransmitter from going into the receptor site, and it will keep it from firing. Acetylcholine molecule receptors are referred to as cholinergic. Acetylcholine has at least 4 functions and binds to at least 4 cholinergic receptors. Acetylcholine acts as a neuromuscular system as an excitatory neurotransmitter, and it allows your muscles to fire. It allows your muscles to work. Acetylcholine acts on the vagus nerve and the heart muscle as an inhibitory neurotransmitter, and it slows down your heart muscle. Acetylcholine is used by the brain in the transfer of memory from short-term to long-term memory. Acetylcholine acts as an inhibitory neurotransmitter to induce you to go to sleep. So as you can see, in some cases it is inhibitory, and in other cases it is excitatory. It allows your muscles to function. It allows memories to go from short-term to long-term memory. But it also blocks the heart muscle, and it inhibits the heart muscle and slows it down. And it also slows down the human body and induces you to go to sleep. Acetylcholine shares some of its receptor sites with other chemicals, and for this reason the receptor sites are named after the alternate chemicals. For example, nicotine binds to acetylcholine sites in the skeletal muscle and autonomic ganglia, and for that reason these sites are referred to as nicotinic cholinergic receptors. Muscarine cholinergic receptors also exist in the autonomic nervous system. Muscarine kills by stopping the heart and paralyzing your intestines. So those acetylcholine sites are called muscarinic cholinergic receptors, because if you arrest those, then you stop breathing and your heart becomes paralyzed along with your intestines. There are 2 types of molecule receptors. Ionotropic, ion-moving receptors open ion channels and allow them to pass through. And metabotropic, metabotropic means change-moving. Receptors activate molecules known as G proteins. The ionotropic movement is fairly rapid. The metabotropic is relatively slow. G proteins bind to the amino acid guanine in its many combinations. These guanine compounds may themselves open the ion channel or be part of a chain of reactions that open the ion channel in turn. That's why the metabotropic is so slow. The ionotropic is almost immediate. There are 4 types of neuronal circuits: neural chains, positive feedback circuits, negative feedback circuits, and oscillator circuits. The simplest neuronal circuit is the neural chain. A sensory neuron is activated by a stimulus, and it in turn activates a motor neuron that responds to the stimulus. That's a neural chain. In a positive feedback circuit, the effect is to increase in activity or keep it going. In a negative feedback circuit, the effect is to stop an activity. In an oscillator circuit, the circuit serves as both a positive feedback circuit and a negative feedback circuit, depending on the circumstance. Most sensory neurons collect data, converge the input through a single neuron, and then diverge the signal when it arrives in the brain. The eye does this. Each moment of sight involves over 100 million receptor neurons that converge the information into the optic nerve, transmit it to the brain so that the signal can be diverged into various parts of the occipital lobe for interpretation, and then we see a picture. The electroencephalogram, the EEG, is a device that reads the electrical impulses put off by the neuronal activity in the brain. EEGs are used to map brain functioning and can be used for sleep, analysis, or to monitor abnormal seizure activity. In the case of a coma, the EEG will detect lack of activity, and that's what we're looking for. And that's what we're talking about. A straight line for it's actually an EEG, and what we're trying to detect its electrical activity in the brain. If there's no activity in the brain, then we refer to the individual as brain dead. And the reason is because they have no electrical activity going on in the brain. Now, normally the brain will, something is always going on, even when you're asleep. You've seen the REM and delta wave sleep. You can see there's always blips going off in the human brain. And normally when somebody's in a coma, we're getting a brain wave of some kind. But if that's a straight line, it means there's no electrical activity in the brain. 30 million people worldwide suffer from epilepsy. Epilepsy is characterized by seizures. Seizures are involuntary synchronized excitation of large groups of nerve cells in the brain. Seizures can be triggered by trauma or injury to the brain, chemical changes derived from metabolic faults, exposure to toxins, mutations in ion channels. Epilepsy is characterized by various types of seizures. Grand mal seizures are the most extreme form of epilepsy. Grand mal seizures are characterized by total muscle contraction followed by alternate jerking and relaxation. The seizures normally only last 1 to 2 minutes, but recovery may take as long as several hours. Petit mal seizures are characterized by losses of awareness of from 5 to 15 seconds. Now, there are lots of different reasons why people may have epileptic episodes. There are individuals who genetically may have an epileptic situation. I had a friend when I was in high school who was epileptic, and he had seizures from time to time throughout his life. Something happened to him when he was in the womb, and it created his epilepsy. But you can also develop epilepsy or epileptic seizures or having seizures from a trauma or injury to the brain. So a blow to the head may cause it. Um, you could be exposed to toxins. There's lots of different reasons that this could happen. Complex partial seizures do not involve the entire brain. The individual may experience an aura, a perceived sensation that precedes the seizures. Certain personality traits often accompany complex partial seizures, but it is not certain whether the traits trigger the seizures or the seizures trigger the traits. Alcohol poisoning. I had a friend when I was up in Montana who would have, he was an alcoholic, and he would drink a lot of alcohol, and if he had an episode where he drank a lot of alcohol over an extended period of time, he would start having seizures. He would start having epileptic seizures, and it was caused by the alcohol. And as soon as he got off alcohol, he stopped having the seizures. This was an extreme case of alcohol poisoning. It was really odd. I was talking to him one day, and all of a sudden, his eyes rolled up in the top of his head. He started tilting his head back, and he went into a seizure. That's the first time I'd ever seen an alcoholic epileptic seizure. And that's what an epileptic seizure looks like. It is just a mass of electrical activity in one area of the brain, as you can see. This is what it normally looks like, and this is what it looks like on I think this is a PET scan. And that is it. And I'll talk to you guys next week.